originally from Mexico City. I did my undergraduate at the Universidad Nacional Autónoma de Mexico, National Autonomous University of Mexico. I went on to do my PhD at the University in North Carolina and a postdoc at one of the NIH institutes, also in North Carolina. Started as a professor at Wayne State University in Detroit. After a brief stint at UNT, I was fortunate enough to be offered to join as a biophysicist in the growing biophysics group in the physics department with a joint appointment in the chemistry and biochemistry department. Several of my family members were scientists. My dad is a physicist and engineer, and so that was always part of my daily life. As I was growing up, there was always toys that had a scientific bent, including like chemistry kits and physics kits and stuff like that. And in those times, the chemicals were not the safest, so that was always fun. As I grew older in high school, I wasn't, I would say, the most focused of students, but I was focused enough that I was able to secure a spot at the top university in Mexico and one of the top in Latin America. So that was just sheer luck, not because of any like really scholastic merits. But after that, I was able to get my bachelor's in chemistry, and friends of the family had always told me, if you want to do computational focused research, you should go into physics, not into chemistry. I was adamant, no, I'm going to be a real chemist. I'm going to work in the lab, right? And then after 6 months doing research as an undergrad in a lab, I decided, no, the smells and this stuff is not for me. So I went into high performance computing. When I was an undergrad in Mexico, UNAM bought the first supercomputer in Latin America. It was a Cray YMP. I applied for a scholarship to be a part of the support people, and so I was kind of like a TA kind of deal where I was working with the researchers at UNAM, providing support for the computational chemistry codes, and so that's where I learned a lot of HPC and high performance computing and stuff, and that has carried me through to now, and that's part of our current research now, so developing codes for high performance computing simulations. So,
Professor, I would like to know what is your current research in, and what are the different departments that you're involved with?
Prior to coming here, I was in the Department of Chemistry, and a lot of the students who were working with me at UNT moved to UTD. The grad students are affiliated with chemistry, and then the postdocs are affiliated with physics. And so this provides for really interesting dynamics and conversations, because we all have different expertise. We have people that are experts in chemistry, people who are experts in chemical engineering, people who have backgrounds in physics, and so it's really fun. The overarching theme is developing tools that allow us to do accurate simulations, and then applying these tools to study systems that are relatively complicated in terms of computational simulations, and so for that we need like really powerful computers. The systems that we're interested in are related to biology, so we're interested, for example, in proteins that deal with DNA repair, or how the DNA is expressed or not expressed, etc., and this has implications related to health, for example, with cancer. So we have projects related to that. We have projects related to just basic understanding of how DNA gets started. We do a lot of collaborations with labs all over the world, and we also develop several different pieces of code. So we have our own code in-house that allows us to do calculations that combine both quantum mechanics and molecular mechanics. If we're trying to investigate, for example, chemical reactions in a big system like protein, or these are called enzymes, we can't solve the Schrödinger equation for the entire protein. We're talking literally of maybe hundreds of thousands to millions of atoms in many cases. And so solving the Schrödinger equation for a system that big is impossible. And so one approach that can be used is a hybrid, where you use quantum mechanics for just a small number of the atoms in your system, and then the rest you treat it more approximately. For example, with a force field. It's called a force field using classical mechanics. And then having these two theories or these two methods talk to each other is kind of tricky. So this has been developed since the 70s. It was work worthy of a Nobel Prize in 2017. So It's a really interesting method. We have done several things related to that, including developing our own code, which is now being developed here. We're continuing the development. And we also participate with other groups, for example, in France, and another one that is multinational, for classical simulations. One is called Tinker, the other one is called Amber. And so we have people that do a lot of programming, we have people that do a lot of simulations of these systems. So there's a lot of different things going on in the lab. Part of the group is interested in more chemical side. We also investigate, for example, systems related to liquid phase simulations. There are many different solvents that are used. One thing that we've been interested in is this particular class of solvents that is called ionic liquids. And so, for example, table salt is formed of two ions, technically, right? So sodium and chloride, right? Those are two ions, and then when you put them together, they form a crystal at room temperature, and they're extremely stable. There are a class of molecules that are also ions, but just because of their molecular shapes and properties, at room temperature, instead of forming crystals, they form liquids. And they have very interesting physical and chemical properties that can be used for all sorts of different things, like carbon capture or catalysis or a bunch of different things. So we've been working on developing tools to study these things in computers. One of our senior students in the group, Madison Berger, published a paper as a co-author where she contributed to understanding how DNA starts the process of synthesis. So whenever the cell is going to divide, you have to copy the DNA, right? The DNA is a blueprint, right? And so this is called a priming. You have to prime the DNA to start copying. There were some hints about how this happened, but we didn't understand it at literally the atomic level. And so we collaborated with a group in the UK, and so they solved the crystal structure, so they actually got the positions of the atoms in space, and we took that model and did calculations to try to understand what are the molecular drivers of that starting process. So that was really exciting. Another project that we've been working on for a while is we developed this method where we combine techniques from biology and bioinformatics, and then computational chemistry and computational biophysics to discover new cancer mutations and then figure out what they do. A recent example that we published last year, we found a mutation on the protein that is related to immunity. Cells have a lot of tools or weapons to fight off infection, right? One of these proteins that are called ApoBecs, or they fall in the ApoBec3 family, is they literally change the DNA of the invading organism. So, for example, HIV. So, for the virus, these ApoBec are involved in HIV immunity. And so, there are 7 different ApoBec3s. And what we found was that in one of these ApoBecs, called ApoBec3H, there's a lung cancer mutation. So, we can go into these cancer databases and search and basically find out mutations that are associated with a particular cancer phenotype that have not been declared before. And then we can use the tools that I was telling you previously to investigate what they do. So a student who is now a postdoc, who graduated in December, he figured out that mutation messes up the protein and it doesn't let it do its job. Could he find out a way to fix the protein? So basically do another mutation, do another change to recover, then we call it recover the activity. And so he predicted rescue mutation, and we collaborated with a group in Canada at the University of Saskatchewan, and they confirmed not only his predictions of what the cancer mutation did, but the rescue mutation as well. This was really satisfying because we were using computers to predict the effects of cancer mutations and also possible ways to fix them, which is kind of cool. We're always interested in working with people who are here to join the group. You can look up our research at Cisneros Res, so Cisneros Res, .utdallas.edu, that's our website, and then my email is Andres, ANDRES, @utdallas.edu. So shoot me an email and we can chat. I've always been interested in providing opportunities and doing science outreach. I've been involved with the Society for the Advancement of Chicanos and Native Americans in Sciences, with SACNAS, for many years. I'm a lifetime member now. At my previous institution, I was a co-advisor for the SACNAS chapter there. SACNAS provides multiple opportunities for students from the diverse backgrounds, so not only Chicanos and Native Americans, but now it's just completely open. One of the main events is a national conference. I've organized several symposia at these conferences. I've also participated in RU experiences, for example, the last one here over this summer. We're always looking for diverse applicants and diverse students, and a strong advocate for diversity in the sciences. So I'm very excited to continue our research here. I've already started at least 3 collaborative projects. Researchers in biology, in chemistry, in physics. The environment is super conducive to great science. I'm very excited to continue doing good science with great people.